Thanks a lot for the introduction, and thank you and Julie for the invitation to come here. It's a really great pleasure and honor to be here with you and share my work and research on microbubbles. I was thinking about what to present, and I was chatting with people today, and I think that people were interested in the work that we're doing with thermogenetic control of T cells and also tracking macrophages. So I did not include those at my talk, but I just while I was waiting for the seminar, I added a few slides at the end. So if there is time, I'll be more than happy to stay a few minutes longer and then talk about these things if there is an interest. But the main topic of my seminar today is going to be about microbubble dynamics, monitoring, and control. So, but before I delve into my research, I just want to say a couple of words about myself. So if you guessed from my name or definitely guessed from my accent, I grew up in Greece where I did my bachelor there in biomedical engineering. I also did a master in medical physics. So, and at the same time, I was about to complete my master, I received a scholarship from the equivalent of NSF scholarship for PhD, and that was the difference. That was international. I could go abroad and do my studies. So I took the scholarship, took the plane, and then went for a PhD at University College London, where I did my PhD in medical physics and bioengineering. And then I went to Oxford for a postdoc, is where I got immersed in the bubble and bubble dynamics at the lab there that is doing a lot of work in this direction. And at the time that I had my first experiments working, there was an opportunity to continue my research in the States. So again, I took it, and then I went to where I also was with you there. I went to in Boston at Harvard and at Brigham and Women's Hospital, where there I started doing introducing bubbles even more and working more with bubbles, but as they're applied to brain diseases. So that's where the two things came together that I will talk today. And then I moved to after a while, I was already research faculty at Harvard and Brigham, but then I decided to go south in Atlanta, and this is where I did all the research that I will be sharing with you today. And also along the way, I met my wife, and we have two boys that it's exactly like that. I'm running behind them, and it's a handful, but it's also fantastic. So I think from my journey, I think I was, as you see, I've been to different places, different cities, different countries, different continents that definitely enrich myself and my research, and also make me, I would say, deeply aware of the importance of diverse thinking, backgrounds, and culture. And as you will see from my lab, all of my students come from different places, different countries, different continents, and I think having this diversity coming together in the lab, I think it's really feel it's a key to our research and also fuels our solutions to healthcare problems. So we're really happy and excited of being able to be in an environment where we celebrate our differences and use them for innovation. So moving to research, I think there is one of the problems that motivates our research is brain cancer. And currently there are no treatments for brain cancer, especially aggressive brain tumors. The current treatments are either highly ineffective or they have significant side effects. And in cancer research, there was a lot of excitement about potentially using immunotherapy. So I will hide Georgia Tech, but maybe we'll be able to see a little bit more. So using immunotherapy for brain cancer can consider as a really kind of the new killer in medicine. But when it comes to brain, they're still faced with many challenges, and there are wide and diverse challenges, but also trafficking and delivery of therapy, of immunotherapy in the brain is particularly challenging. And if you think about that, if you have an intravenous administration of a therapeutic agent, so it has to go a very long journey. It has to travel in the vasculature, identify the vessels of the cancer, extract from the vessels, then has to penetrate through very dense extracellular matrix and structure, and then it has to engage or bind to the cancer or the immune cells as immunotherapy. Right? And there is a lot of good science and evidence indicates that if we target the immune cells of brain tumors that can constitute up to 30% of the mass, you might be able to come up with something that's meaningful and potentially impactful. So, but these are like also many challenges, and perhaps one of the challenge that has attracted of most interest from the community is the blood-brain barrier. That is considered really the major barrier for the penetration of therapeutic agents in the brain, and that's for many good reasons. So, we need our brain to maintain homeostasis. We really need to have specific balance of analytes and molecules there. But this at the same time creates enormous obstacles to delivery of drugs. Brain tumors, there is a long discussion in the field about brain tumors appear to be leaky, but detailed research, both in the clinic and preclinical, indicates that there is leak in some places, but still it's highly heterogeneous environment that ultimately the delivery remains as a major challenge, especially, for example, for glioblastoma, where the tumor recovers all the way around the main core that usually is removed by surgery. So still is a major challenge. So clearly there is technologies that will be able to overcome this challenge and improve the immunotherapy, the delivery of and trafficking of immunotherapeutic agents can have a very big impact in this medical problem. So switching now to the focus ultrasound, which is really the topic. So microbubble, when combined with focus ultrasound, can potentially provide some meaningful solutions to this problem. This is what I would like to focus. So this technology, some of you may already know in the audience, has like two key components. First is the focus ultrasound that I don't know if this plays. So it really allows us to focus the beam through the skull in the brain. So, and I want you to appreciate this is a human skull, that you can appreciate that this is totally non-invasive, it's targeted, and can go deep into the brain. So these are like really key components of this technology as opposed to any other technology. And compared to optics, you can go deep. Compared to radiation, there is no damage on the path because the energy is focused primarily in the focal region. So the other component, which is really the core of my talk, is the microbubbles. So microbubbles have been originally discovered as imaging contrast agents. So they're lipid most of the time cells. They can be polymers or other structures, but the clinical grade right now are lipids. So you can inject them, and as they circulate everywhere, they will end up in this focal region here. So in this region, when they experience the pressure wave, they will start vibrating, and they might vibrate. As they vibrate, they might exert radial forces. They can have radiation force push to the vessels. They can create streaming patterns, same way as you stir tea or coffee, or they can implode. And all these forces can be sensed and felt by the endothelial cells, in that case, in the vasculature. And there is like a good evidence that they may make tumors more amenable to therapy. Okay, so what I would like to talk to you today is I will elaborate a little bit on the methods that we developed to monitor and control the bubble dynamics, and this is with a goal to promote, if possible, specific interactions. And also, you'll see that the truth and the elegance that we currently have might allow us to gain mechanistic insights on the different interactions, and this could further be used to potentiate therapy. Also, some of the aspects will be used for diagnosis, and I will allude to that. Okay, but let me say, how do we, the principle, and this is really the fundamentals of bubble dynamics. How do we monitor and control microbubble dynamics or acoustic cavitation? I will use more microbubble to avoid the word cavitation, which also includes the formation of bubbles. In my work, we already have the bubbles there. We don't form the bubbles, okay? So really what we care is we really take advantage of the information that is contained on the acoustic emissions that are generated when the microbubble vibrates. So there is this vibration. And there are two specific components that weigh this. As you can see on the left-hand side, we have the excitation pulse. It can have a certain frequency. So when the pulse interacts with the bubbles will start vibrate. And then when they vibrate in the mode that we call stable cavitation, stable microbubble dynamics, they will start generate harmonics, ultraharmonics, and subharmonics. So this information can be really critical, reflecting of the type of oscillation. If we increase the pressure, the bubble might collapse due to the inertia of the environment. And this will essentially create a baseline shift in the frequency domain data that I'm showing there. So this will create what's called broadband emission. So overall, you can also, needless to say, this is like highly nonlinear effects. You can see only from the acoustic characterization, you have F node there, and you have a wide range of frequencies in the output, which is definition of a nonlinear system. So it's a highly nonlinear system at the same time. So it's this content of the acoustic emissions, right, that we can measure them with a hydrophone. We tend to call the passive cavitation detector, so that we try to infer what the type of oscillation that is taking place. Okay, and using that, we can monitor, localize. If we have beamforming methods, we can start controlling, have specific oscillation, and then with this way we can kind of promote desired type of oscillation. And then there is the last step is we then do correlate. That's what we do so far, the type of oscillation with a specific bioeffect, and then we use this bioeffect to promote therapy. Okay, that's the broader thought process in the cavitation detection monitoring, how it can be used for basic research and at the same time for applied research. Okay, so I just want to emphasize why this is important. Okay, so it's really, in fact, essential. Okay, so for every therapeutic intervention, we really need to achieve a certain level of microbubble activity. We need to achieve a certain level of force exert, right? So we really want to push the pressure to the point that the bubbles have certain vibration. So this, this will allow us to ensure optimum outcomes, okay? There's also a nonlinear phenomenon. When it goes to the brain, we don't always know the pressure. So nonlinear, without knowledge of the pressure, is very hard to tune the pressure setting. So, but we really need to do it in order to achieve the effect, okay? So safety matters a lot as well. So you cannot really start decreasing the pressure, because you can start having off and on-target adverse effects. If, for example, in the discussion that I will talk about, which is immersed in the brain, so we have seen that when we have stable oscillation, strong stable oscillation, right, we can disrupt the blood-brain barrier, and this can still be safe. But if we start increasing the pressure and what we start seeing inertial cavitation, we can start creating holes in the brain. Because you can start damaging the brain, essentially. So it's really important to be able to push the pressure as much, but not cross the limit, because it's safe. So it's also reproducibility is crucial as well. So if we think about microbubbles, every microbubble can be different. We can have like significant difference in the cell. This might impact their oscillation. We can have differences batch to batch. They have different kinetics. They have limit in FDA. Also, the focus ultrasound system has instabilities. External ultrasound is reflected. This will impact the impedance. It will create fluctuations in the output pressure, not to be reliable at the same time. Most important, nobody has really, many people have explored, but mostly we're trying to understand it, but there is a biological variability involved. So if we do the same experiment with the different animals, this variability might be important. So we really need to monitoring and controlling the bubble dynamics is really critical for like really achieving this kind of like, efficacy, safety, and reproducible treatment. So it's really, really critical to tame the force that is contained within the microbubble dynamics towards therapeutic or diagnostic purposes. So in my lab, we have developed two different methods to do that. In one, we use the amplitude of the excitation pressure. So essentially, we have the ultrasound transducer, and we build closed loop mechanism. So what we're trying to do, we're trying to maximize certain acoustic emissions or harmonic component amplitude. And then we just close the loop, and then we just send the pulse, what's the amplitude? Is it lower than a threshold or higher than a threshold? And we try to adjust the pressure, okay? And that's happening in a closed loop function. And we also have used frequency. And then we're interested in frequency because microbubbles has a mechanical structure characterized by resonant effects. So can we use frequency to explore the resonances and try to identify operation regimens that potentially lead to new or more effective treatment strategies, okay? So let me start from the first one, okay? So that's a little bit, I have to say some of my slides are very dense, but I really wanted to share with you our, rigorous experiments and also the insights that we gain. So I will flash them as I go along, so it's not too much. So here is like, a system, the closed loop control system that we developed, that the goal is maximize the harmonic level, which is indicative of strong, stable oscillation, but at the same time, minimize the wideband emissions, okay? And the system has three components. First is the harmonic level. So what harmonic? We have many harmonics. We have subharmonics, ultraharmonics. So what harmonics are we gonna use to tune our system? So we did like a bunch of experiments for different pressures. and we use the changes in the blood-brain barrier permeability using MRI, and we just try all the possible components and did the linear regression. We find which one produces the best data, the best correlation, which means for a certain level of harmonics, we can predict and promote certain level of broadband disruption. And we did find that third harmonic was provide to us the best results, okay? And you can see here from the top, as you increase essentially the harmonic strength or the pressure, you can have the ultra harmonics kicked in and broadband emissions kicked in. So this becoming to the region where we want to avoid. So that's one component. The second component is the control algorithm, okay? So the control algorithm is really a proportional controller that is based on a cavitation threshold model. So what the controller actually does is it follows, you can see, we connect pressure to harmonic emissions level, and here is from harmonic emissions level to K-trans. So this is a transformation that is taking place. So when we increase the pressure and the harmonic emissions follow the pink line, the controller feels confident. I'm following a predicted trajectory, so it increases the step. So it goes fast to a certain level, okay? If it goes off, it slows down. It's the same as if you're in this room and you see where the benches are and you turn off the lights, you can navigate and go fast, even if you don't see the benches because you know what's going on. But if you move the benches and you don't know what's going on, you're gonna go slow. So it's exactly what the controller is doing. And you have this cavitation threshold, and it allows us for training and stability, okay? And the third and really important component is the controller also tracks the microbubble dynamics. So because the microbubble go in and they have 5-minute circulation time. So you can envision, right? So I have here a harmonic level or there. This is really depends on the concentration of the microbubbles. If the microbubbles start clearing out, the controller will think that doesn't have enough pressure. So we'll start increasing the pressure to a point that we'll get to a regimen where the bubbles will collapse. So we really track the kinetics and we tell when the controller should be on and off, depending on the amount of bubbles that we have in the focal region. And that's kind of like an additional level of controlling and monitoring what's going on. Okay, so this is track the microbubble kinetics. So with all these components, so we just track all this at the same time. And to track the kinetics, essentially, we send this pulse. We see that doesn't have any impact on the K-trans, but it's still detected. And then we just send this pulse every time we send ultrasound pulse. For the controller, we send a small, weak pulse, and we track the kinetics. You will see how this looks like. So this is our control data. So you can see this is the pulse that tracks the kinetics, and it's activated when it passes, the first ramp after the point, that point, which is roughly like 10 dB threshold, and then it will remain on until we have a 20% decay of the concentration. And then you can see we just chosen 2 different levels, and you can see the controller was able to achieve those levels within the window that is on. After that, it just adopts the last pressure and continues constant pressure, okay? And you can see here we're able to have like quite significant, very small onset of wideband. This histogram shows how many broadband events we had and how strong they were for all the sonication that we tried. You can see here that there is quite few present. So what was interesting, what was surprising to us, the first surprise from this research is that we observed that if you see the gray data there, this is just using constant pressure. You can see that there is a wideband emission. All of it happens at the beginning, happens at this point here. So the controller is able to adjust the pressure and avoid this kind of strong inertial cavitation events that happen when the concentration of the microbubbles is very high. So it is able to maintain. So it tries to rise up to a certain threshold. If it detects broadband emission, drops. Okay, so we still have broadband emission events, but they're significantly reduced. Okay, and then you can see here the 2 different levels. We can see we fall within our calibration data. So the controller is able to achieve the levels that we prescribed to it and being able to change the blood-brain barrier permeability in an exposure-dependent level. Okay. So we felt like confident with this data. So let's try to see something therapeutic. So just use an antibody, which is immune checkpoint inhibitor. This is like an antibody that essentially amplifies immune responses. It acts mostly on the T cells, but there's also evidence that it might act on the tumor microenvironment. So we feel like let's try it. And there are like few key observations here. First, as you can see here, it will still have this exposure-dependent increase. So we go from low to high exposure, it increases. But what was also interesting to us is, if you see here, this is the antibody, and you see the concentration drops. This is the control animal in this category here. Here is the macrophages. Here is the immune cell. So you can see we have high antibody in this region that the immune cell is low, and we have no antibody in this region that the immune cell is high. So essentially, if you don't do something, you might still have some antibody, but does the antibody get where it's supposed to be going? That's we don't know. But when we apply this control method and focus ultrasound with microbubbles in general, you can see we have very uniform distribution, very high distribution of the antibody, and we see significant interaction with the immune cells of the tumor microenvironment. Okay, so that's kind of like give us the evidence that if we are able to employ these methods, we will be able to have higher concentration and also lower gradients of the drug in the tumor microenvironment. And we try to do a survival study. And we did see in the blue line, as you see, we see like a small but statistically significant improvement in survival. It's very small, but if we have this in the clinic, it's going to be amazing. It's going to be equivalent for FDA approval, although it's very small. That's so devastating, these diseases. But when we also collected the animals that saw like completely tumor eradication, we just reinjected humans, we challenged them, and we did see that the tumor never grew. So essentially, we kind of provide evidence that when we have like the immunity, we develop anti-cancer immunity in these animals, which is very interesting and fascinating that we don't know why, but still being able to improve the penetration of the drug or disrupt the blood-brain barrier can lead to cure in some senses of this disease, which is quite exciting. So, and I just want to mention, in parallel to this work, there is clinical trial that's going to try to explore this interaction. So that's a work that we have published some time ago. So one key component that this controller has, and I mentioned, and this is unpublished work, is that it is what we call reactive. So it tries to reach the threshold. If it reaches the threshold, it stays there. If there is a broadband emission, it will drop. Okay, so essentially it reacts to the microbubble collapse. So the question was like, the student Henry Lee asked is, can we start like, thinking about predicting the response? And that's where like, predictive modeling coming. So what we kind of like start working on is we still have the controller, the normal controller. This is this part here is this controller going in the normal route. But at the same time, we feed the data to a neural net. And this neural net, it takes all the frequency components, the microbubble kinetics, even the location, if a disease, we can put any data we want there. And what it does is, it tries, based on this data point, tries to predict if the next indication will have wideband emissions. If the next indication has wideband emissions, then it overrules the controller and tells the controller, no, you will not change the pressure. You will reduce the pressure if its intention was to increase the pressure. Okay, so essentially it provides, an advisory role. The controller tries to get to the point, but if the data based on like a huge data, the beauty of that we were able to do this project because Henry, that was working on this PhD, was like a wonderful guy. He was helping everybody in the lab, and I encouraged you to do that. And a student was saying, "Oh, I want to deliver this drug. Can you help me?" Yes. Another student, "I want to deliver this drug. Can you help me?" Sure. And they was able to collect thousands of data sets from all the experiments. So because of that, we were able to have this. We trained the network with about 60 animals. Every animal had 5 targets, and every target had 120 locations. And we use only the previous one to predict the next one. So essentially, we have like 50 to 60,000 data points to train this network. It's really important. That's one important aspect. And the other aspect is you really need to have data in your distribution. So you really need to have data with wideband emissions. You really need to have all the possible permutations to be able to trust the predictions of the model. The model cannot predict something that doesn't know. But if it knows the distribution of your data, which be all the data I saw before, it might be able to start sense and detect differences, small differences in the data, and then give you information. Okay, so that's the key new component. And you can see here what we tried to do 26 and 30, but because already the wideband emission was too low there, so we pushed it all the way up to 36 decibels. So that's kind of like what you should have broadband. So that's where we say that we use the controller. You can see here, it tries to get to 36, but when it starts predicting, kind of like drops. So that's the drops of the controller. So that's the data. So how do we compare this? So we want to compare this response. So we try three different aspects. So the first aspect is, okay, let's take the Pmax, the maximum pressure the controller reaches, and then use this as one group. So we say the extreme condition. Then let's use the average pressure that the controller, the machine learning informed controller, ended up using. And we use this, and these are constant pressures. And we also use the closed loop control that I shared with you before. So I have 4 groups that we compare with. Okay. So, and you can see here, they're like, when we compare, there are like some interesting observations. First of all, if you don't have the controller, you always have an overshoot. So when the bubble come in, you're going to have wideband emissions, essentially, right? So, and if you can see the mean, it's quite close. You can see, the maximum has a little bit higher. But, these that are non-significant because you have high variability in the non-control cases, essentially. Okay, but you can see, they can achieve pretty much similar response in terms of harmonics. There also is like, when we take the deviation, you can see that the control data have much more deviation because they try to maintain and successfully maintain certain level of harmonic emission. Okay, and then when we go to compare the K-trans, which is the blood-brain barrier permeability, you can see the P average and the P max. They just pretty much all over the place, but they did have high signal. I believe this is mostly because due to overshoot. But when you have the closed loop and the machine learning controller, you have again a little bit, slightly more reproducibility, and they stay in this level. So if you take only this data, you feel like this doesn't work well. Okay, admittedly, you have similar response in harmonics, but you have like, much better permeability. So then we just start thinking, okay, let's take a look at the content of the broadband emissions and see what you have here. Pay attention to this blue line. So we have 1,000 sonications. We only had 1 wideband emissions out of 1,000 sonications. And if you compare with all the other controllers, of course, the Pmax had the most, and you see they have way better response. But here it has way much more damage. Okay? And you can see the P average and the closed loop controller have similar response. Okay? And here is the events. So what we also did, we got this data, and we say, let's feed them to the network. Will it predict the case where we had wideband emission? And in fact, it was able to also predict pretty much most of the wideband emission. So even if we don't have the closed loop, if we just use the machine learning network, it will also be able to give us very good prediction about the wideband emissions and what may have prevented that. Okay, so next is, okay, let's go to safety. Let's take the tissues out and then let's analyze them. So we did, a bunch of analysis. I will highlight here only the GFAP, which is astrocytes, and you don't want astrocytes called astrocyte activation. So if you see the machine learning, which is here, did not create any astrocyte activation, whereas the P average only, which is the average value, created significant astrocyte activation. We didn't see any microglia activation differences. We think maybe that's not the time point. We might need a little bit more time that we sacrifice the animal, but we still, you can see by 2 major fundamental metrics of assessing damage in the brain, the machine learning controller has no major, use any major, markers that are relate to damage. Okay, so now that was the most surprising finding to us. Okay, so we really started this project for safety. Let's make sure that we increase the safety. But what we realize is as we go through, we can see here the star. This star here indicates for the different conditions, this is all the non-machine learning controllers. When is the time that we have the first broadband emission? Okay, and if we get and try to define what's the treatment window, what's the safety, and you can see this is pretty much close to 1.2 megapascals. When we have the machine learning, essentially we more than double the treatment window, because essentially the machine learning, when you have normal controllers, it's going to try to go high. When it gets broadband emission, will drop, right? But the machine learning is pushing. When it predicts, it stays there, and then it's pushing again, and then it's pushing again. So essentially, you have like and also it's sensitive to microbubble concentration. If the concentration drops, the controller is not responding to a time point or rigid threshold. It adapts to the data all the time and makes predictions. So this really more than double the treatment window for safe, right? No wideband emission, okay? And if you can see here, this is the star again. You can see here in the K-trans, it relates to much higher K-trans for safety. I remember that I told you before that the K-trans was higher, but they had broadband emissions data. What if we take the data that don't have broadband emissions? What I want you to think about this data is that this is the normal cavitation data. What machine learning does is skew the distribution, essentially. So we just skew the distribution of how the normal data will produce wideband emissions if we increase the pressure, and we kind of like, we'll have this behavior. It just happened to be linear for this data. I don't think it's linear. Everything is highly nonlinear, but happened to be linear in that case. But essentially, you can see like a shift in the distribution that pushes the safety lower, but increases the blood-brain barrier permeability. That was really, really, we did not expect this finding at all. And so the next question was, what are the implications? Does it mean anything to this? Does it mean anything? Can we do something that we couldn't do before? Okay. And then one more step. So the question before that, we asked, can we scale it? So all the data that I showed you before was with mice. So let's go to rats. And I will explain, we have many good reasons to go to rats. So we just went rats, and not only rats, we just also have tumors. We also have tumors in the previous data, and it's sensitive to tumors as well. The controller makes different decisions. So you can see here the behavior is pretty much the same. So you can see he's able to predict, use the wideband emission to greater extent, and also predict the wideband emissions in the animals that they had wideband emissions. So the controller is really, we had to tune it, but the principle and the framework is scalable. Okay, the actual numbers need to be tuned because you have much different physical, the phenomena are weighted differently, right? So, but the framework, it appears to work perfectly well at a larger animal, so it's scalable. So then next we said, like, let's see if we can deliver nanoparticles, okay, which is used for drug delivery. You can see here this is the brain tumors. You can see this is the K-trans. The first is control. Second is we use the standard blood-brain barrier disruption, which can be considered borderline safe, a little bit wideband, and then we use the machine learning controller. And you can see a remarkable improvement in the delivery of nanoparticles. These nanoparticles are 45 nanometers. So you can see that we were able to achieve delivery that we couldn't be able to achieve with p average. So this is like really the first key finding and the first implication of like expanding the treatment window, essentially. Okay, and the next question that we ask is, can we, because this is a tumor, we transfect the tumor to express GLUC gene, and this GLUC gene is a secretable, generates a protein that is secretable, and we want to see, can we increase the detection of cancer-soluble biomarkers? Okay, so this could be used for non-invasive characterization. It can be used for a bunch of reasons, right? You can see we have same tumors, we have no FUS, p average, which is a little bit, above the safety threshold. You cannot go above that, for example, in the clinics. And then we have the machine learning controller. And what you can see was that was remarkable. So we were able to achieve much higher protein. That's not a big issue. We have seen this data in mice. I didn't present it today. But what we were able to see, we were able to improve the secretion of the circulating tumor DNA. This is the first study that shows that. You have seen circulating tumor DNA, but if you see the histology, the brain is full of blood and damage. So if you have seen proteins, but circulating tumor, which is the holy grail, essentially you are able to detect mutations. You are able to genotype tumors if you are able to detect circulating tumor DNA. Even if you have treatments, you might be able to detect resistance to treatment. So that's really a major finding. And we went to great scale. We didn't succeed in mice. We went to rats. In rats, when you see liquid biopsy, usually you take compromised mice, because if you use immunocompromised, it doesn't clear. So we did immunocompromised, it didn't work because we had huge background. When we use immunocompetent, which is the hardest model to do liquid biopsy, this is where we see, and this is the most clinically relevant. We start seeing this remarkable, I would say, response to that. So that's kind of like this data demonstrate that using statistical analysis into decisions about how to tune the microbubble dynamics using the acoustic emissions can create new opportunities for diagnosis as well as for treatment of brain diseases. So I think that will be like, exciting and make all the whole liquid biopsy field a little bit closer to being applied to a wide range of diseases. So I just want to emphasize this is not something that is out there. So we were lucky enough to collaborate with Dr. Graham Woodruff, who is a neurosurgeon at the University in Baltimore, Maryland, and he tried this experiment with InsightX system in patients with glioblastoma. You can see at the bottom left corner, you can see that established feasibility in humans. You can see in the middle, which is H&E, demonstrate that it's safe. And also on the right-hand side, which is a fluorescent dye that neurosurgeons inject in order to identify the tumor and resect the tumor. When he resected the tumor, he was able specifically to collect data from region of the tumor that was enhancing in any case, region of the tumor that was not enhancing in the middle, and region of the tumor that was targeted with focused ultrasound and was not enhancing before. Goes back to what I said, that not all the tumors are leaky, and was able to show that when we apply ultrasound, we have improvement, but also the acoustic emissions can be used to predict the disruption, as it shows this data, which plots the subharmonics as a function of relative contrast enhancement. So this data can potentially, this framework I described, if you have enough data, and I want to emphasize that every patient is nowadays, you have 64 targets. Every target has hundreds of indications. So you have 6,000 points for every patient. So even with 100 patients, you're going to have 600,000 points that you will be able to train this network and essentially translate this work to the clinic. So I want to move to the second part, and that's like, how do we use frequency? Okay. So, and that we are really interested in the frequency is an important parameter. No discussion about that with respect to ultrasound. It's defined the field by the frequency. Okay, but also bubbles are characterized by resonant effects, and then that's kind of like we really are interested in seeing whether we can exploit that. But it's very hard to study resonant effects in vivo, right? This is bubbles. Their dynamics is not a single bubble, is also impacted by the environment. You have vessels, so it's a couple dynamic. So how do we come up with something that's meaningful? And to do that, we start from mathematical modeling. So we build a mathematical modeling of microbubble dynamics in vessels that were surrounded by porous media. And our goal here is we were interested in identifying conditions where we can maximize the wall shear stress. So the stress that is applied to the wall by the microbubble. And there are many reasons to do that. And most important is a lot of endothelial cells are very sensitive to wall shear stress. Okay. So first we wanted to validate a little bit our model. So we use two different types of bubbles. One is rigid, which is the albumin, and the other is more elastic, which is the lipid cell. It's a commercial product. We isolated the size, same size bubbles, and then we just did the modeling, and we did see that the bubble that is the definity, which is a lipid cells under the identical conditions, should exert much more stress, or should vibrate much more. So how do we prove that that's the case in vivo? Acoustic emissions. Same size again. So you can see here now when we have the definity, which is the more compliant, have much stronger sounds. It's the same what will happen to my voice cord if I start screaming. They will vibrate much more, right? So this kind of provides an evidence that at least at the first stage, that what the model says that the bubble that is with label will vibrate more, we have this evidence, okay? And we were able to see also that it has an impact on the blood-brain barrier as well, when we try the two bubbles, identical conditions. So that's kind of indicates that bubble is extremely sensitive. Identical mechanical index as well. Bubble properties matter. Okay, cell properties matter. It's not mechanical index that matters only. Okay, and you can see we also did the analysis of the tight junctions. So you can see the definity tight junctions are downregulated, which means the blood-brain barrier has been disrupted much more effectively using the more elastic bubble. Okay, but this really has direct implications towards the kind of vision and thought of how can we have therapeutic bubbles. Bubbles that have designed for therapy, not for imaging, and have been repurposed for therapy. Okay, so and then we just did we decided to go with a lipid, which had most effects, and we did a frequency sweep. And you can see on the left-hand side is a standard resonant plot. So you have different frequency and different radius. So you can see kind of the peak there. This is a coupled resonance in the vessels. So what was interesting here, I wanted to emphasize So if you see here in this case, if you go 0.5 and 1.5 frequency, the displacement is the same. So the bubble delta R over R is identical. You can see if you go to the y-axis. But if you go and see the shear stress, essentially is 2 to 3 times higher. You see from 0.5 to 1.5. So what this happens? This happens because the shear stresses depends on the bubble displacement, but also on the bubble velocity. So at higher frequencies, the velocity is higher. And then it's getting lower because then you don't have enough displacement. So it's the combination of those. If you go off resonance, the displacement becomes smaller for a given pressure. But if you're just above resonance, you might have the maximum potential force, exertional force. And this is for 2 micrometers. And then we just, okay, let's isolate 2 micrometer bubbles and see what happens. Okay. And yeah, so here are, I had the annotations there. Okay, so much higher wall shear stress when we go just above the resonant frequency of that system. And then we just went in vivo. And then what we did is we did like RNA sequencing. We just harvested endothelial. And what you can see here, you can see significant expression of markers that are associated with leukocyte migration, right? So it's essentially inflammatory markers, and they're very critical for facilitating immune cell infiltration, essentially. And all of those are very sensitive to shear stress. So this kind of like provides evidence, right, that maximizing the wall shear stress essentially translates to much more upregulation of inflammatory markers. Okay, we just used one of those intercellular cell adhesion molecule, which is very critical for immune cell transmigration from the blood to the brain. And we also did protein analysis, and we do see that this molecule is indeed expressed. So it's not only at the transcript level, it's also at protein level. But also, what is also very interesting, and also another surprising finding, is that we didn't see any correlation with the blood-brain barrier disruption. So while we have an increase in the inflammatory markers, the blood-brain barrier disruption pretty much remains unaffected, if not reduced. So this has 2 major implications. So the first implication is, if we go to low frequencies, essentially, we can disrupt the blood-brain barrier, but not have inflammation. This can be like really important for if you want to deliver drugs in diseases where we don't want to have inflammation, right? This is really counterintuitive. In ultrasound, the mechanical index is what defines the safety. Here we say we're going to go low frequency to be more safe. And on the other hand, it can be used for immunotherapy. So if we can increase inflammation, we might improve the immune cell trafficking. This is exactly what we did. We collaborated with a doctor, is behind the camera there, Okada from UCSF, who is a pioneer in CAR T-cell therapy in glioblastoma. And we were able to show here, this is our theory, we have E-selectin, P-selectin upregulation, and ICAM, which are key molecules to facilitate immune cell transmigration in the brain. And we were able to see robust CAR T-cell trafficking in the brain tumors. In this case, we use EGFRB3 CAR T-cells in SB28 glioma. And we also, that's the good thing of collaborating with people that have more extended experience in immunology. We also took the CAR T-cells and we confirmed that they expressed the receptors that engage with ICAM and P-selectin, which provides an additional evidence that viewing ultrasound was like really facilitated and amplified the transmigration of immune cells to the brain. Okay, so really, putting everything together, I think mechanical stress can be exerted by the microbubbles, can potentially be used to improve CAR T-cell trafficking in brain tumors, can improve antibody delivery, can improve nanoparticle, can improve detection and liquid biopsy. If you are able to tune your system at the point where we'll be able to get the most out of it. Okay, and that's the summary that I had. So I think tuning the amplitude frequency can really be used to refine and also define the treatment window, right? We are able to come up in a new way to think about what the treatment window is to support therapeutic, or in the treatment window for diagnosis is even higher, because you might don't have the disease, right? And also, just as a highlight as I go along, can inform studies to start engineering bubbles for therapy, okay? And it can improve the delivery of a wide range of highly potent therapeutics, antibodies, nanoparticles. You can start thinking about gene delivery because you have nanoparticles, or CAR T-cell, which are like engineers cells that can target very specific drugs. And also it can augment the secretion of ctDNA. This is the holy grail in liquid biopsy, and the brain is even more important because you cannot just put a needle and collect samples whenever we want. Okay, so that's what I had. Just want to thank and highlight, the work Henry Lee. Here he just did all the work with the microbubble dynamics. Victor, Henry has mechanical engineering. Victor is mechanical engineer, but he feels like biomedical engineering research is the best thing that happened in his life. Changing disciplines, he really appreciated it. And he did all the work with nanoparticle, developing all the assays for the liquid biopsy cDNA. And also Yutongo, she did all the modeling and the mechanical engineer, but she be developed with support and collaboration from Han Zhongzhou, the RNA sequencing and isolation, and also Grim Woodruff and Heide Hogan, and also by our funding. So this score can be supported by the National Cancer Institute, the Focus Ultrasound Foundation, and also Ian's Friends Foundation. They were like quite relevant for that. So I think I don't know how much over. I'm a few minutes over time, but I think I'm good.
Maybe we'll start with the questions here. Anyone has any questions?
What was that DNA thing in the brain that you have to do that quant that you can like upregulate? What does that do? I don't know what it is.
What do you mean DNA? Or is it protein? Or
it's like a CT.
It's DNA. So it's called circulating tumor DNA.
Yeah.
So that was we wanted to compare both protein and gene. So we transfected the cells to express the G-loop gene. So this is a reporter gene. And that's what, because we knew the sequence and also we knew the protein, so we want to compare. You have to think protein, every cell produces thousands of proteins, but has only a couple of gene copies. So it's a very huge difference in scale in terms of like products. Also, it changed the background. So this is like a reporter gene. We haven't done any gene, oncogene or gene related to therapy per se, but we're working on that right now. After we established the principle, we're gonna try to identify genes that they are clinically relevant and potentially more important, druggable. Genes that if we can target with a drug, it might lead to responses. Did this answer your question?
Mostly, yeah.
Okay.
I don't think I know enough.
Okay, sure. Sure.
Any other questions?
Yeah, I had a question. So going back to the ctDNA, so there's really impressive results with the machine learning model. And how many points did you say that was trained on, that particular model?
50,000.
Okay. So do you think that when that increases, is there like a diminishing returns and how much that can increase that?
I know for sure there is a bottom limit. So machine learnings, and that was the criticism that was receiving in like 10 or 20 years ago, is not better if you don't have high number of data. Right. If you don't have data that will cover the distribution of the behaviors of your system. So in that sense, the more data you have, the better it becomes. The challenge is to train it, but after you train it, here is real-time decision, because the model, it takes time to train it, but once it is trained, it's real-time. So I feel like the more data you have, I don't see why it will not be better. So I don't see an upper limit, but I see a lower limit. You really need to have all the possible scenario in the system that you're studying to be able to trust what it tells you. And still, it's not perfect. We had the sensitivity was, we want to be conservative, so we want to have low false negatives, but it was 95%. But we didn't care about false positives. If the model predicts that there is a wide band, but there is none, we didn't care because it's safe. This was 55 to 60%. Now we're talking with colleagues from the computer science, and they try to bring all the arsenal of machine learning, and they can boost further up to 85, 90, 90%, both of them. So the more data you have, you can improve the sensitivity and the specificity of your model. And then it's also what will also give you, if you have a very well-tuned model with 90% sensitivity and 90% specificity, I didn't include this data. What you can do is you can go back and ask the model, what are the key variables that you used? What did you weight more? And what we found there, we found ultraharmonics is more, and there is a group that has developed control with ultraharmonics. We find harmonics is important, and we have developed harmonics. what was surprising, we did find that it predicted much more wideband emissions in the tumors compared to healthy brain. Nobody has ever reported that, and that was very surprising to us because this might also start having implications for diagnosis, right? And you can also, by feeding data at hard broadband to model, also predict even more wideband emissions when it comes to tumors. So it also gives you the opportunity to understand the environment and the biology, maybe, but also what are the conditions underneath and why we have these wideband emissions. It's kind of like, makes us ask questions that we wouldn't be able to ask before, right? And also gain insights on bubble dynamics in the more complex conditions that the acoustic emissions in isolation couldn't tell us. I think I had it in my, okay. Why this? Okay, other questions?
I have a follow-up question on the machine learning controller.
Yes, go for it.
So my understanding is that it predicts whether the